Modern dentistry relies heavily on mechanical force. When a tooth is lost, the standard replacement involves driving a titanium screw directly into the living jawbone. Researchers have attempted to bypass this mechanical damage by using stem cells to engineer new dental tissue. However, building a complex organ like a tooth from scratch remains biologically unstable and expensive. There may be a more direct route. Deep within the tissue of the human jawbone lie microscopic structures called tooth primordia. These are biological seeds, already perfectly positioned to grow. Humans naturally cycle through exactly two sets of teeth, deciduous baby teeth and adult permanent teeth. Yet the DNA instructions required to produce a third set remain present, though dormant, in our genetic code. Regenerative dentistry is shifting focus from building new teeth in a lab to triggering these existing architectural blueprints already buried inside the jaw. Tooth growth is driven by specific biological pathways. The most critical is the bone morphogenetic protein, or BMP, signaling pathway, which acts as the body's natural molecular accelerator for tooth morphogenesis. To keep this growth in check, the body produces a protein called USAG1. Its primary role is to suppress that growth signal. This diagram illustrates the interaction. A glowing BMP signal attempts to connect with a cell membrane receptor to trigger growth, but it is physically blocked by the USAG1 protein, which brings tooth development to a complete halt. Researchers at Kyoto University tested this by removing the USAG1 protein in mice born with congenital tooth loss. With the protein removed, the BMP signal was finally able to successfully bind to its receptor. The arrested tooth germs responded immediately. The dormant cells activated, generating the complex structural growth necessary to form entirely new, healthy teeth. This suggests that toothogenesis, the inability to grow teeth, results from an overactive molecular suppression system rather than a lack of physical building materials. But there is a biological risk. USAG1 also regulates a separate network called the Wnt pathway. If researchers neutralized the protein completely, they risked triggering unintended skeletal mutations throughout the body. The team needed a neutralizing agent that would target USAG1 with surgical precision. They developed and tested 5 distinct monoclonal antibodies, searching for one that could stop tooth suppression without interfering with other skeletal processes. This 3D topographical map shows the result, antibody number 37. By targeting this specific 10 amino acid segment, the antibody blocks BMP interference while leaving the rest of the protein structure available for healthy Wnt signaling. To determine if this would translate to more complex biology, the team moved trials to ferrets. Like humans, ferrets possess both deciduous baby teeth and permanent adult sets. A single systemic dose of antibody 37 produced a significant result. A new incisor emerged directly alongside the mature permanent tooth, forming a third dentition. These results demonstrate that by using engineered molecules to isolate highly specific biological pathways, researchers can safely direct the body to regenerate its own organs. This breakthrough points toward a cell-free approach to dentistry. Rather than implanting foreign hardware or attempting to control lab-grown stem cells, the goal is to use targeted molecules to signal the body to use its own resources. Monoclonal antibodies are already used to treat arthritis and certain cancers. Utilizing this established framework for dentistry is safer and more cost-effective than traditional tissue engineering. Trials in pigs and dogs are the next step before human testing begins. Researchers will use genetic biomarkers to identify candidates who carry the specific genes, like EDA1, that make them likely to respond to the treatment. This potential therapy relies on a critical biological prerequisite. The patient must have a foundation of healthy jawbone and viable, dormant primordia to act as the starting point. Preserving that foundation starts with current dental care. Managing gingivitis and maintaining jawbone density are essential steps to ensure the biological matrix remains viable for future molecular interventions. While titanium implants remain the clinical standard today, these molecular results in animals suggest a future where we can tap into the human body's own latent regenerative power.